Hello, welcome all of you. We are going to discuss a new chapter, new concept, that is the RNA editing. So, all of you know what mean by the RNA, ribonucleotides, and editing means to change something in a ribonucleotide. Okay, to change in RNA, that is the ribonucleic acid. So, RNA editing has a great importance in eukaryotic system. Okay, so there is a huge difference in between the post transcriptional modification and RNA editing. Okay, so as you know, what are the various methods or mechanism of the post transcription modification? Within that, 5 prime capping is there, 3 prime polyadenylation is there, okay, splicing is there. This is a mechanism which is followed after the transcription process, but there is a difference in between this post transcriptional modification and actually the RNA editing. Okay, so within the post transcriptional modification there is a nascent mRNA or nascent RNA is get uh, processed, and there is a 5 prime capping is adding to the 5 prime position, and 3 prime polyadenylation is occurring at the 3 prime position. In the splicing there is the intron splicing. One is the there is a two codons regions in the whole genome, that is the introns and exons. Exons are the coding regions, introns are the non-coding regions. So we have to remove the introns, which are the non-coding sequence regions, which are present in the eukaryotic system. Okay, so this is the post transcriptional modification. So RNA editing is a somewhat different in both in the case of post transcriptional modification. Okay, so what mean by the RNA editing? So RNA editing, it is an uh, process other than splicing that results in a change in the sequence of RNA transcript such that it differs from the sequence of DNA template. Okay, it means it is a changes in RNA nucleotide sequence. Okay, so in the case of splicing, in the case of R5 prime capping, in the case of 3 prime polyadenylation, there is a no change in ribonucleotide sequence. Okay, in this case, RNA editing, in the case of RNA editing, there is a change in ribonucleotide sequence or the RNA nucleotide sequence, except the intron splicing, 5 prime capping, and 3 prime polyadenylation. Okay, this process is a very rare, and the definition is clear as the DNA template sequence is differ from the RNA transcript. Okay, at the time of DNA transcription, there is a DNA, and DNA get from the DNA, RNA is get synthesized in the process of transcription, but DNA remain as it is. Okay, there is no change sequence in the DNA. Okay, so only change in the RNA sequence. Okay, the first considered as a bizarrely now recognized widespread RNA editing process, which information changes at the mRNA level. It reveals by the situation in which coding sequence of the an RNA differs from the sequence of DNA from which it was transcribed. So basic, what is the fundamentals, what is the need of the RNA changes? So eukaryotic cell have a number of work at a time. Okay, so within a particular single cell, eukaryotic cell requires number of proteins. Suppose that number of proteins is 100. So within a 1 minute, within a second, cell have to prepare number of proteins. Okay, 100 proteins in a 1 minute. So its gene are more active than that of the prokaryotic case. Okay, so RNA or cell have to maintain the synthesis of these proteins. So in this case, one gene can responsible for the synthesis of many proteins. Okay, as you know, the one gene, one protein or one enzyme, one gene hypothesis. Okay, so this is a previous hypothesis which maybe at the after the research. Okay, so one gene can code more than the one protein. Okay, so that protein is responsible for the various metabolic functions, various metabolic processes within the cell. So this is the very important, that is the one gene is responsible for the production of various proteins. So how this occur, what, how the one protein is responsible for the production of many proteins? So the DNA sequence is same, but when the RNA editing is done, at that time the framework for the coding regions, framework for the amino acid is get changed due to the RNA editing. So there is a change in the protein sequence. Okay, so after the transcription there is a what, translation. So amino acid is adding by the tRNA. So tRNA recognizes the codon region at its anticodon region, and there is a formation of new protein. Okay, so this is what, why the RNA editing is important. Okay, so we can see now the our presentation. So editing events may include insertion, deletion, or base substitution nucleotide within the edited RNA molecule. So you can see that in this case, insertion, deletion, or base substitution of nucleotide within the editing RNA molecule is occur. RNA editing occurs in the cell nucleus or the cytosol of the cell, or within the mitochondria or plastids. Okay, also occur in few chloroplast gene of plants, at least few nuclear genes in mammals. Okay, RNA editing is reported in protozoa, plants, mammals, not in fungi, prokaryotes, nuclear, mitochondria, chloroplast, and viral RNAs just like mRNA, tRNA, and rRNA. Then the discovery of RNA editing is transposon mitochondrion. Within that this RNA editing firstly discovered by the K. Stott and Simpson. Okay, there is the two general types of the RNA editing. That is the one is the base modification, and another one is the insertion and deletion. Okay, so what mean by the base modification? So what is the main types or changes will occur in the RNA editing? That one, first one is the addition, second one is the deletion, third one is the modification. Okay, so what happens in the uh, addition or in the deletion? U insertion or deletion is seen. After that, in the base modification, A to I means adenine is get converted into inosine. That is a new type of base, or the variant of the nitrogenous base, double stranded mechanisms in the virus and human genes. Okay, this is the main type of RNA editing which is occur in the uh, RNA editing. Okay, so function of RNA editing by deamination, which is the first process, that is the deamination, which is the enzyme, one of the most important enzyme, that is the double stranded RNA dependent deaminase, which is responsible for double stranded RNA deaminase is responsible for the RNA editing. So this is the function of editing by deamination, mRNA translations, RNA structure, alter protein binding, pre-mRNA splicing, RNA degradation, RNA replication. Okay, so RNA editing, there is a one diagram of how the RNA get edited. Okay, you can see the transcription. Okay, this is the first of all there is a sequence of DNA. After that there is a process of transcription. The DNA gets sequence, DNA sequence get converted into the RNA. Remember that the sequence of the DNA is a coding strand sequence. That's why the sequence of RNA is same as that of the DNA strand. Okay, so there is no confusion. The sequence of DNA is a coding strand. That's why the sequence of the RNA is same like that the coding strand. After the transcription, there is a uh, editing. Okay, so there is addition of A. Okay, and this A is replaces by the U in the case of the A get complementary with the U in the case of RNA. Okay, so translated of the edited RNA that leads to the formation of new protein. Okay, editing by the insertion deletion. Editing through the insertion deletion of variation found in the clinoplasm from the mitochondria tryptosomia brucei. Because of this involved in the large fraction of sites in the genes, it's sometimes the pan editing to distinguish the typical editing or the few sites. Means this is the guide RNA mediated RNA editing. Okay, so this is the diagram of the RNA editing. You can see the DNA. After that, transcription of DNA to the pre-edited RNA. Okay, guide RNA gives the template for the editing. So there is a binding of the guide RNA. What mean by the guide RNA? We can see. So guide RNA is an sequence, RNA sequence, which is responsible for the editing, and which is a small nucleotide sequence of the 40 to 70 nucleotide and complementary portion to the mRNA. Structural elements anchor information part of oligo. You tell base pairing of guide RNA with unedited RNA gives a mismatch region which are recognized by the editing machinery. Okay, so guide RNA has a sequence of the 40 to 70 nucleotide which is a complementary to the portion of mRNA, at which the RNA editing is done. At that portion there is a complementary sequence with the guide RNA, and that guide RNA recognize that particular complementary sequence and edited that portion of the mRNA. So RNA editing mechanism, you can see insertion and deletion. In the case of deletion, there is a guide RNA which is pairs with a complementary base pairing with the mRNA, and you can see there is a in red UUU sequence is there. Okay, so in the case of deletion, there is a removal of UUU, and there is a binding of this 5 prime to 3 prime direction of mRNA. Okay, strands of mRNA. In the case of deletion, there is a cleavage in between the in between the phosphodiester bond of the RNA, and cleavage is occur by the endoribonuclease. There is a addition of U. Okay, after that addition of U, there is a binding of this U, those strands. Okay, so this in this way the guide RNA can catalyzes the RNA editing. Okay, so RNA editing is a catalyzed by the multi-protein complex. Okay, so this multi-protein complex contains or the proposed or the made up of endonuclease, exonuclease, tutase, RNA ligase, and helicase. These all are enzymes are comes under the multi-protein complex, which is responsible for the RNA editing. Okay, editing by the deamination C to U. Okay, this process there is a removal of NH2 group. Okay, by the double stranded RNA. Deaminase, there is a adenine has a NH2 group, or cytosine has a NH2 group, which is the removal by the this double stranded RNA deaminase protein. Okay, or deaminase enzyme which is responsible for the removal of NH2 group. Okay, so there is a conversion of C to U. Okay, this you can see this is the editosome complex. Okay, and this editosome complex is responsible for the RNA editing. Okay, you can see here the guide RNA is also there. Okay, and in lower portion there is a mRNA. This is the anchor DNA, and this is the editosome complex. Okay, this is a multi-protein complex which is responsible for the RNA editing. Guide RNA is responsible for the to recognize the sequence of mRNA at which the RNA editing is to be done. Okay, this is the example of FOB gene. Okay, which is the DNA strand. After the transcription, there is a pre-mRNA strand. There is in the case of intestine, RNA editing changes to CAA to UAA, causing the stop instead of a gland. Means there is a further transcription or further translation is get stopped due to the CAA. There is a formation of C get converted into U. That's why the formation of this is UAA. This is the termination codon. That's why the protein synthesis gets stopped. After that, the liver. In the case of no RNA editing, uh, apolipoprotein B100 is protein is get synthesized, and in the case of intestine, due to the what RNA editing. Okay, that is the insertion conversion of C to U. That is the formation of stop codon. That is a UAA, and that causes the stop the translation. There is a formation of apolipoprotein B48, means 48 base 48 protein, 48 amino acid is there, and in apolipo B protein there is a 100 amino acid is there. Okay, effect of C2U RNA editing in the human ApoB gene. This is the which gives a clear idea about about the RNA editing. Okay, so apolipoprotein, and here is the same, that is the unedited RNA in the liver, and edit unedited mRNA in the intestine. Okay, so this is the mechanism of how to convert the C into the U. There is a NH group that convert into the carboxyl group, oxide group. RNA editing. Okay, this is a chloroplast in mitochondria. C to U. Okay, in the some case U to C is also happen, all coding regions. Okay, so after the A to I, this is the unusual mechanism that in the RNA has NH2 group which get removed by the deaminase enzyme, and there is a formation of inosine from the adenine. Okay, and the enzyme is called as the adder means double stranded RNA dependent deaminase. Okay, so this enzyme is responsible for the conversion of this adenine to the uh, inosine. Okay, so this is all about the RNA editing. Okay, significance of RNA editing. It is essential in regulating gene expression of organism. RNA editing mutant was reported strong defects in the organelle development. Deficiency causes the disease. It is a mechanism to increase the number of different proteins available without a need to increase the number of genes in the genome. May help to protect the genome against some viruses. Okay, so this is the significance of RNA editing. Main significance is the to get the number of proteins from a particular genes which are already present in the cell. Okay, so there is no need to extra genes to for the production of more protein. Okay, so this is the main theme behind the main significance, main function of the RNA editing in this case. Okay, so everyone, I think everyone has understand this RNA editing, how what is the process behind that. Main basic three sequences or C three mechanism is important, that is the addition, deletion, and the modification, that is the C to U, A to I, that is a inosine. These are the important points within this, and why is important, this also very important in this case. Okay, so thank you all of you.